Yeah, I already did. So we should be on there in a moment. Okay, he just emailed me back and said he got it. There. Okay. All right. Do you want to?
Oh yeah.
You're gonna start.
Hi everyone. Thanks for coming to Elizabeth's defense. Elizabeth joined us in spring of 2015. She comes to us from Nova Southeastern University with a bachelor's and a master's degree in science in marine biology. From there, she examined potential impacts of incorporating artificial reefs within the boundaries of marine areas to enhance fish production and aid in habitat restoration. So we've maintained her trajectory here with this study on artificial reefs. She moved to Tampa to pursue her education at USF and began as an OPS employee in my lab, where she assisted on deepwater horizon oil spill work. While at USF, she has worked as a science mentor for the oceanography camp for girls. She is also an active adjunct instructor at the University of Tampa, teaching marine biology and environmental science courses. Today she's going to give her presentation on her thesis, Fish Communities on Natural and Artificial Reefs in the Eastern Gulf of Mexico. Her academic committee is composed of Pam Halleck-Mueller, Chris Stallings, Will Patterson at the University of Florida, who is watching online, and myself, Cameron Hinckliffe. Thank you very much.
Okay. All right, everyone, thank you so much for coming. So as Cam said, my thesis is going to be differences in fish communities on natural and artificial reefs throughout the Eastern Gulf of Mexico. So before I get started, I just wanted to give a little bit of information on artificial reefs, especially since I study them at my time at Nova and continue to pursue artificial reefs here. So a little bit about their history. We've been using artificial reefs for a variety of reasons for quite some time. We've seen them as far back as about 400 years used by the Japanese specifically to enhance their fish stock. So definitely a useful tool when it comes to fisheries. In the past 4 to 5 decades, however, that's really where we've seen that particular field in marine science grow in America, in Canada, in Australia. And when it comes to artificial reefs, there is a huge variety in regards to the construction of these particular reef habitats. So we've seen anything from sunken vessels and ships to cars, sunken loads of tires, oil rigs, lots of different types of materials. But nowadays, really the most effective material that we have found to use is concrete. And even within the realm of concrete artificial reefs, you still see a huge variety. There's still a lot of different types of structures. There's a lot of research out there about how the complexity of these structures is going to influence their ability to achieve whatever goal is set for them upon deployment. So with that being said, lots of different reasons why we deploy artificial reefs. They're used for a variety of things. So anything from expanding existing habitat to targeting a specific species to protect, whether it's an endangered or threatened species. A lot of you are probably familiar with the approach to supporting those recreational activities like diving and fishing, used a lot for those. And the big one that we're going to talk about today is enhancing our fisheries production overall. So there's a lot of research out there because we've been using artificial reefs for quite some time. And these particular projects that have been done over time take many different approaches. So you have research out there that's comparing natural and artificial reefs. You have research that's looking specifically at an artificial reef's ability to attract fish versus produce new fish. And you have research that focuses primarily on their use as a tool to enhance commercial fisheries. So lots of research, lots of variety within the field of artificial reefs when it comes to their construction, why they're used, and how we study them. So because there is so much out there on artificial reefs, we wanted to come up with some research questions that were not only going to help maintain and solidify the knowledge that we already have of artificial reefs, but we wanted something, or to come up with something that was going to be innovative and help push that field forward. So the first research question that we came up with was, how does community structure differ between natural and artificial reefs at different levels of taxonomic resolution? And this is really important because we hypothesized that when most people look at artificial reefs versus natural reefs, they're going to tell you these are different habitats, there are different species present. While that is true, if we were to broaden the level of taxonomic resolution by looking at the corresponding families of those species present or looking at their functional role within the community, we may end up seeing more similarities than we previously thought with these particular habitats. The second research question we wanted to ask, again, expanding our existing research on comparing those two types of habitats, was how does the environment and the actual location affect the community structure? So is it just the physical artificial reef structure that's driving those differences, or are there environmental variables that need to be considered when it comes to the particular types of species represented at these habitats and the location? Where are you in regards to water depth, in regards to latitude? There are all these different components that we believe are going to also influence the structure in the community that we see. The last main research question we had was, how do artificial reefs respond to fishing? And this question we were able to answer because of the nature of a huge portion of our data, which I'm going to get into momentarily. But sub-questions that we can expand on with just that one research question, how do artificial reefs respond to fishing? There's a lot of management approaches that we can take in answering that question. So we wanted to look at not only how do they respond to fishing, but can they be used as a management tool when it comes to the fishing industry, and can they actually supplement productivity and biodiversity for our marine environment? So the data, I was so lucky. I had huge contributors to my data set that I built, which was a feat in and of itself, if we're being totally honest. I've got over 15,000 observations on reefs. It was a massive data set, a massive undertaking to get it all together, make sure everything was consistent. So I'm going to break down where all the data sources come from. But I definitely would not have been able to do a project at this scale if I didn't have all of these sources. So I'm super grateful for all of that. The first data set we got was strictly artificial reef data from the northern Gulf of Mexico. And this was given to us by Dr. Patterson and his lab. These were artificial reef surveys performed specifically by ROVs, or remote operated vehicles, off reefs that are deployed off Panama City, Florida. His surveys took place from 2004 to 2010. He did quarterly surveys, so they were repetitive. His data, super unique relative to the other data sets that we had, because these artificial reefs he surveyed, they were deployed by FWC in 2003. However, FWC did not release any of the coordinates to these artificial reef sites. So they had 27 reefs deployed, and this gave Dr. Patterson and his lab a really unique opportunity to monitor how fishing can potentially affect these reefs. So they divided up their reef sites into 3 groups. They had 9 control reefs, 9 tagging reefs where they were going out and tagging fish, and 9 what they called fishing reefs. And in 2007, in the summer, FWC published the coordinates for the fishing reefs only. And they continued to survey all of these reefs through 2010. So they were able to do a lot of research on the impacts of fishing in that particular region and on artificial reefs specifically. So again, their data set provided us a really unique opportunity when it came to developing our research questions. The second data set that we acquired was natural reef data from the northern Gulf of Mexico. This was our largest data set that we got. It was given to us by Chris Gardner at National Marine Fisheries Service in Panama City. And his data was collected, or their lab's data was collected off Panama City and Apalachicola, Florida, from 2005 to 2015. So this is 10 years of data. It's a lot. They performed their surveys a little bit differently. They were using baited stationary video surveys, which are definitely different from the ROV. ROV performs transects. The stationary video gets plopped down. They run it for 20 minutes. bring it back up. That's how they collect their data. Then they analyze the video feed. So a little bit different collection methods, but again, nice contribution to our growing data set. The third data set that we got, natural and artificial reef data from the West Florida shelf. So it's a nice combination of both reef types. This was given to us by Ted Schweitzer with FWC, and these were the CMAP surveys that they were doing on the West Florida shelf from 2008 to 2015. And they used the same techniques as National Marine Fisheries Service. So it was the baited stationary video. Really important before we get into all the analysis, the methodology for collection of this data. Different, ROV versus stationary video. So what we did after we built this big data set was we normalized all the count data so that we didn't run into any issues, have any discrepancies with any of our results from our analysis that we were going to do. And then lastly, for the second part of our project, we needed to get that environmental data. So Cam has a nice contact. His name is Arnaud Gross. He's now with University of Miami. He helped build this large environmental database for the Florida Restore Act Centers of Excellence program. And they house it in what's called the Gulf of Mexico Research Initiative Informative and Data Cooperative, or GRID-C. This is an online database that you can go onto, select portions of the Gulf of Mexico, and pull out environmental data. Within this data set, there are things like sea surface temperature, bottom salinity, depth, nitrate concentration, phosphate concentration. There's a huge variety of data there. And the collection methods are all going to be different as well. Their sources are coming from research facilities, universities, cruises performed, buoys that are deployed. There's a huge variety in the collection methods as well because the amount of environmental data and the span of that environmental data is very great. So we went through the GRID-C database, pulled out what was relevant to our particular study as far as environmental factors are concerned, and tacked that into our ever-growing database. So this is a map of all the reef sites that we had. So as you can see, you've got some more natural reef sites versus your artificial reef sites. The data has just been collected for a longer period of time. But just to give you an idea of the region that we're going to be talking about today, so we consider this whole area part of the northern Gulf of Mexico, and this is going to be our West Florida Shelf region. Okay, so the first method that we chose to use was an ordination method, and this was applied to our first research question, comparing those artificial and natural reefs at different levels of taxonomic resolution. So the ordination method we chose was a principal coordinate analysis, and this allowed us to really explore and visualize any dissimilarities within our data. And for those of you not super into statistical analysis or modeling, I'm sure many of you have heard of a principal component analysis, or a PCA. That's a really common test to be run on, relatively sizable data sets. The main difference between those two tests really comes in with the dissimilarity or the distance metric that you use. With a PCA, you're limited to Euclidean distance, which isn't the best for environmental data or zero-inflated data. So we chose a PCOA. That way we were able to choose our dissimilarity metric that we wanted to use. We went with the Bray-Curtis metric, which much better for biological data. So after you run a PCOA, you basically get two types of output. You get a Cartesian coordinate plot that plots your observations in a two-dimensional space, and you get a vector biplot. These are produced using a type of eigenanalysis, and the latter, the biplot, is just as equally as important as the Cartesian coordinate plot that I'm going to show you here in a minute. Shrinking your data into two-dimensional space, especially when you have as large of a data set as I did, super challenging. That's one of the reasons why we went forward with the different levels of analysis as far as the taxonomy goes. But the biplot that I'm going to show you with each of these coordinate plots really representative of the coefficients that are driving the placement of those principal coordinates that are plotted in that Cartesian coordinate plot. Another thing to note about the vector biplot is the length and the direction of the vector is indicative of the magnitude of the variance and the coefficient that's driving that variance within that data set in that particular part of the plot. So, results for our principal coordinate analysis. This was our first plot that we got. So this is the species level. We weren't expecting to see a whole lot of similarity here because talk to anybody who's in marine science and familiar with the Gulf of Mexico. One, they're going to tell you the West Florida Shelf and the northern Gulf of Mexico are completely different habitats, and you can't compare the two. And same goes for artificial versus natural reefs. They're two different. So one thing I do want to point out with all of these is the axes and the percent variance explained by each axis. So we were never really expecting a huge amount of variance to be explained because of the size of our data set and how we were trying to shrink it down into a national plot. But at this it was significantly low. You're talking about 18% variance explained by this plot for all the data, which is very, very small. But there was a couple of things that kind of jumped out at us with this plot. First of all, you have a relatively clustered area of your artificial reefs in the northern Gulf of Mexico. The other thing we noticed was the black plus signs here. Those are artificial reefs on the West Florida Shelf, and we don't really see any sort of pattern with them. They're kind of just everywhere. What we attempted to do, which unfortunately I wasn't able to, was find the deployment dates for those artificial reefs to see. It was kind of our thought that maybe they were deployed a couple of decades ago and they have assimilated more into the natural environment. But unfortunately, that was a question we weren't able to answer. The blue dots are the natural reefs in the West Florida Shelf, and the purple are the natural reefs, or I'm sorry, blue are northern Gulf of Mexico, natural reefs, purple are natural reefs on the West Florida Shelf. So there is a little bit of a gradient between your northern Gulf of Mexico sites. But again, not a lot, very little gradient, or very little variance explained here. And you do see a little bit of overlap with your two natural reef sites. So if you look at the biplot associated with this, we had so many species that most of them, you can't even tell what they are. So the couple that stuck out to us were a red snapper over here, kind of indicating that maybe they're having some sort of influence on our artificial reefs in the northern Gulf. You had vermilion snapper up here, a couple little grunt species stick out, gray snapper. Not a whole lot of information from this, but the red snapper is going to become very important later, so just keep that in mind that we saw that all the way out here at the species level. Then we go to the family level, and our plot cleaned up a little bit. Looks much nicer. So our variance explained combined with the two axes went up to about 40%. Which for a data set of 15,000 observations, I'd say is pretty decent. You still have this cluster of artificial reefs in the northern Gulf. You then have a lot of overlap with your two natural reef habitats down here. And then you have this little cluster up here, which kind of has a little bit of everything. Mostly natural reef data, though. You have a couple observations on artificial reefs. So if we look at the biplot, tells a much cleaner story than the previous PCOA. So again, we saw red snapper pointing towards those artificial reefs in the northern Gulf, and now we have Lutjanidae, which is our snapper family, pointing towards this cluster again. This cluster that has a little bit of everything, but is a little smaller, we saw the jack family pop out, Carangidae. And then this big blob of natural reef habitat with a little artificial mixed in, had a variety of families. We see our Labridae, Sparidae, Hamulidae is in there. You have some Serranidae, your sea basses specifically in there. A couple different families that are sort of similar in the marine environment, kind of all pushing towards this big cluster here. So from this point forward, we wanted to go to the functional group level, anticipating that it was going to give us a more clear picture than we saw at the family level. However, the plots are extremely similar. The variance explained is almost identical when you compare the two, right around 40-ish%, 42%. You still see this cluster of artificial reefs in the northern Gulf. You still have this cluster of mostly natural, but definitely some artificial in there. And you still have this gradient down to this cluster here. It almost is as if the plot kind of just rotated a little bit compared to the family level. But what we cared about was the biplot when we ran it at the functional group level, because of the way we classified our functional groups through those Atlantis functional groups that I was mentioning earlier. So snapper again, making an appearance. You have a couple other species over here, demersal fish, some large reef fish, but the longest vector for this quadrant is going to be your red snapper. And that's, again, leaning towards those artificial reefs in the northern Gulf of Mexico. Carangidae family was parent in the family level, your Jack functional group, which is an identical group. So anything that's in the Jack family is going to be in the Jack functional group. Same cluster, same representation. So your Jacks are driving the majority of the variance in that data. Then over here, where at the family level we had 6 or 7 families that were sticking out, relatively obvious. Now we have something called small reef fish. And upon these results, we went back. I went through what families actually fit into this small reef fish group in Atlantis. Turns out all the families that we saw pointing to that same cluster at the family level fit nicely into this small reef fish functional group. So the plots are very similar, but it does give us a little bit of information. So perhaps the functional group level not really going to give you a tighter picture as far as the comparison between the two reef sites or the two locations, but it does provide you some additional information that we found to be quite important. So what we decided to do after we got the results for the family and the functional group level is we wanted to see what was going on with these particular clusters of points. Now, it is important to mention that in a PCOA, your points on this Cartesian coordinate plot are not sites. These are all individual observations. So that's why there's so many and not, we had 200, 300 reef sites perhaps, way more observations than that on this particular plot. So what we wanted to do is we wanted to pull out the clusters that we found here. So we had our Jacks, snappers, and then we went to the functional group, and we had 3 of them here. We had our red snapper cluster, our Jack cluster, and our small reef fish cluster. And we wanted to see, just kind of get a visual of where these were located in our study area. So for the family level, we plotted our Jack and our snapper groups. And what we found was a little bit interesting, and kind of what led us to our second method to answer the rest of our questions, is that regardless of the location, what you're typically seeing is the majority of observations that are closest to shore, typically dominated by your Jack family. The northern Gulf is a little bit harder to recognize because it's just a narrower shelf. But then as you move offshore, you kind of shift a little bit in your dominant species, and you have much more snapper activity the farther you go. Same thing here off Apalachicola, and if you were to really zoom in on this area here, you definitely have a lot of Jack observations close to shore. The fewer observations you have farther off onto the shelf are dominated by snapper. So even though the shelf is smaller here, you tend to still see a very similar gradient at each location between those two families and their distributions. So red snapper and small reef fish, there were tons of observations for those guys. So I put them on 2 separate maps so you could actually see what was going on. So red snapper found throughout the Gulf of Mexico. That shouldn't come as a surprise to anybody in this room. And then our small reef fish, again, that functional group includes, almost a dozen families. The distribution is going to be, very spread out throughout the Gulf, not going to be any super specific consistencies in regards to which location they dominate. You see them throughout the entire study area. So upon receiving our PCOA results, now we had to answer some of our additional research questions about how the environment is affecting this distribution, how the environment is affecting community composition at these different locations, at these different types of reefs. And so we chose to pursue a multivariate regression. The method we chose was a generalized additive model. And what we did was we pulled out our dominant families and functional groups, and we built a GAM for each one. And The reason we chose a GAM is because this additive model approach allows you to visualize your variables in a nonlinear form, so it allows a little more flexibility. And because it's multivariate, it allows us to analyze several environmental factors at the same time. So for this part of the study, we expanded, we went with our reef observations and our environmental data as one data set into the GAM. So just a little bit of background on GAMs. I won't bore you all too much with it, I promise. This is a basic GAM equation here. You have your response variable, which for us was our fish counts, and you have your predictor variables, which was our environmental data. This is a list of our predictor variables that we decided to use. So our depth, bottom salinity, bottom temperature, bottom dissolved oxygen. We chose bottom versus top because that's where our reefs are sitting, on the bottom, not on the surface. We also wanted to look at the turbidity and the current speed in the area, the chlorophyll concentration, the distance from shore. As we already saw, that was an important factor from our PCOA maps that we pulled out. And then we used what we call eastings and northings, which were converted from our latitude and longitude. And the reason that we did that is to get a better sense of the spatial autocorrelation between these different observations and these different reef sites. So what that conversion essentially did is it allowed our model to look at the spatial autocorrelation with a higher level of accuracy, because the eastings and northings calculation look at the distance between locations on a sphere rather than in a two-dimensional space. So it gave us a little bit more of a clear picture. And then these last four predictor variables here are our sample month, our sample year, and then we had two coefficients that we created ourselves. So these are binary data. So is it an artificial reef, or is it a natural reef? And is it on the West Florida Shelf, or is it in the northern Gulf of Mexico? The reason that these are a little bit different as well is that they're considered parametric data. So we don't use any sort of smoothing function. Now, when I say smoothing function, GAMs are going to have smoothing functions. They're also going to have link functions. So basically, the distribution we chose for our GAM was a negative binomial distribution. And we chose this particular distribution over a gamma or a binomial or a Poisson distribution specifically because its ability to accurately depict variance in biological data or data containing lots and lots of zeros, which is what we had. The link function here is important relative to the distribution. So the link function helps to specify the relationship between the predictors and the mean of that distribution. And then we have our smoothing functions. Our smoothing functions are what, represented by that S sub n, what we apply to each of our environmental variables that allows them to have a nonlinear relationship. We use two different kinds of smoothing variables. So for the environmental data, we chose a thin plate regression spline, and then for our eastings and northings coefficient, we chose what is called a tensor product. So tensor product was only used for those eastings and northings specifically because you can combine, you can put covariates together with a tensor product, and it will be able to smooth it regardless if the units are identical. So it's a useful tool when you're trying to look into that spatial autocorrelation component of the model. And then lastly, we use something called a REML method for our selection method that has something called downweighting. And this was really important because the model itself will adjust your smooth terms if they are not significant to the model or to the data. So basically we'll penalize them and select them out of the model rather than having to do those AIC tests where you look at your AIC and then you pull a variable, and then you look at your AIC and try to get the optimal AIC. This allows us to kind of get around that whole process. So these are what our GAM results looked like. I'm going to put up all of our smooth terms here so you can see what types of environmental factors are really affecting these groups. And then I put up our parametric coefficients that were the most significant. So those are your smooth terms here, all environmental factors. So everything I listed off before. The asterisks are indicating that the p-value was less than 0.05, so a very significant p-value. And then the little point here, the period, that's telling you the p-value is somewhere between 0.05 and 0.1. So still significant, but the asterisks are going to be more significant factors. So those are your parametric coefficients that I was mentioning. So none of these had smooth terms associated with them. The main things that I'm going to be pointing out, which I think are really important, especially when it comes to our conclusions, is this spatial autocorrelation coefficient here. As you can see, it's one of the most significant terms for the family Lugenidae and their distribution, as well as whether or not it's on the West Florida Shelf or the Northern Gulf, and whether it's artificial or natural. So prior to going into this part of the project, I was told by many people that those two locations, Northern Gulf, West Florida Shelf, are different. You can't compare them. And the natural versus artificial, different, can't compare them. They're going to make the communities completely different from one another. So for the family of Lugenidae, that was very clear. We had extremely low. This was the lowest p-value a GAM will spit out, too, by the way. So it can be 2 to the negative 6. It could be 0. So extremely low p-values for both of these. And the spatial autocorrelation is very important as well. We also see that bottom temperature, really, really low p-value as well. And again, lots of environmental variables can potentially be affecting this particular family. When we moved to Pharyngidae family, we got a little bit of a different story. Again, spatial autocorrelation, lowest possible p-value along with depth. You also had a significant p-value with distance from shore, which if you recall my map of our distribution of snappers and jacks, distance from shore was definitely something that was influencing their distribution. And then if you look over here, these p-values, while still significant p-values, are larger than what we saw for family Lugenidae. And they're larger than what we see for depth and for that spatial autocorrelation term. So yes, they're influencing the distribution of our jack family, but spatial autocorrelation and depth are having a greater influence than the reef type or the location. Then we moved on to our small reef fish functional group. Lots of variation here again with our reef type and location. So artificial reef or not, definitely having a very significant influence. Location, still significant, not as significant as reef type. Again, spatial autocorrelation, very, very, very significant, as well as a slew of environmental variables. This group is really diverse. They have lots of different feeding guilds in that particular group. There's small predators, there's small herbivores, small detritivores, they all fit into there. So we were kind of expecting to get a lot of significance here because there's such a variety in that particular group. But again, this part's pretty important. Artificial reefs or natural reefs, definitely super important for this particular group, but the location, not nearly as significant as the reef type and the spatial autocorrelation. And then for our last group, red snapper. Location, artificial reef. Very, very important. The other most significant term is our spatial autocorrelation. You do have a couple environmental factors here, current speed, turbidity, bottom salinity, a little bit from chlorophyll here. But this functional group, red snapper, and our family, Lugenidae, or our snapper family, were the only two groups where we saw equally significant impacts from reef type and location. The other thing is that all of our groups showed equally significant impacts from that spatial autocorrelation component. The other thing that's really important, really interesting that we found with the red snapper group was 2005 to 2010. This is lots of Will's data here, Dr. Patterson. So the yellow was before the release of the coordinates to the public for fishing. And then 2008 through 2010, fishing was allowed on a third of those particular reef types. What we saw was that these years have highly significant p-values, And then theoretically, upon the release of the locations of those reefs, the p-values increase in size, no longer providing significant sample years, or as significant sample years, I should say. We do see a little bit of on and off as far as the significance of years after 2010. I think it's worth mentioning that Will's data didn't extend beyond that particular year, so this data is all from the other two data sets that we had. Additionally, this year, 2010, if you all recall, that was the oil spill year, deemed the year that the lionfish invaded. So there's lots of different factors that kind of could be driving some changes in the significance of the sample year for red snapper. But again, that is going to require a lot more research, and I'm not really sure if it's going to be possible for you to distinguish the factors of Deepwater Horizon versus lionfish since they happen in the same year. I don't think you'll be able to say it's definitively caused by one or the other. And again, location, reef type, really important. So one of the main takeaways that I was able to pull from our GAMs, which I was not expecting, was this concept of connectivity and how important it is. So each of our groups that we ran a GAM for, the most consistent lowest p-value among all groups was that spatial autocorrelation component. And that is a huge testament to how important connectivity is between reef sites, reef types, locations in our little stretch of Gulf Coast Florida waters. So just a basic definition of connectivity, really just linking populations among larval exchange, eggs dispersal, juveniles, adult movement, as well as adjacent and distant habitat. So how close a habitat is can help determine the connectivity. But for this project, I really dove into evolutionary and demographic connectivity because those kinds of connectivity are really important for management. So your evolutionary connectivity focuses on the level of movement among animals that helps to maintain a really healthy level of genetic diversity within your populations. This is obviously very important when it comes to biodiversity conservation and restoration of habitat. We know populations need a healthy amount of genetic diversity in order to, have that longevity that we want them to have. And then your demographic connectivity really focuses on how the movement of those animals affects the growth and decline rates of a particular population. And relative to management, obviously that's something we want to study because that is going to help us make proper policies surrounding sustainable fisheries, help us manage particular areas that are maybe economically important for tourism and things like that in our marine habitat. So this was an unexpected surprise, but makes sense after we did a little research on our results. So just want to conclude with first our principal coordinate analysis. So what we found, we supported our hypothesis. At higher levels of taxonomic classification, you had much more similarities between artificial and natural reefs. You had much more similarities between the West Florida Shelf and the Northern Gulf of Mexico reefs. Out of all of the combinations of reef type and location, where we saw the most overlap in each level was the natural reef habitats in both locations, regardless of that taxonomic resolution level. And lastly, the groups that really stuck out to us from this part of the project were our red snapper. Whether we were looking at the species level, they were still very obvious there, all the way up through the functional group level. Those particular species really driving some variation on those artificial reefs in the Northern Gulf of Mexico. And the jacks and the small reef fish really focusing on community composition differences and similarities between natural reefs in both locations, Northern Gulf and West Florida Shelf. And with our GAM, after we had taken our main groups and run it through the GAM, we looked at 4 different groupings, 2 families, 2 functional groups. We saw that it really wasn't just the reef type that changes the distribution or the community structure in general. It's not just the location that's having an impact. And within each group is affected by different environmental variability. Different environmental drivers are changing distributions depending on what group you're looking at. The spatial autocorrelation term, like I mentioned, That was the only one that was consistent through every single group that we looked at, and consistently extremely low. And that is really where we pulled that concept of connectivity from, how important that is to the reef environment on the West Florida Shelf and the Northern Gulf. And that, again, has some definite implications for management and conservation of biodiversity long term. And then with the red snapper situation. So after those coordinates were released, the sample year for red snapper, the significance of the year changed. We saw a drastic reduction in the significance, so an increase in that p-value. And what we wanted to do with this, just a little side note that we did to make sure that we weren't misreading those results, is we selected out those reefs that experienced fishing for the whole survey time for Dr. Patterson's lab, and we ran a t-test on before fishing and after fishing, and we saw a significant p-value between comparing the two. So we know that definitely influenced that particular group. Now, to say that it's the only thing that influenced them, obviously based on all the results we got from our GAM, that's not the case. But it did have an effect on that particular group of organisms. So there's a couple things that, if time was no object and time was no object, I would love to do to expand on this research. So I'm going to share those with you. One of the big things that is really, really important to artificial reef research is this concept of rugosity. And rugosity is basically how we're defining the complexity of these artificial reef habitats. So the surface area to volume ratios, the height, the width, how many nooks and crannies are in there. If you read about artificial reefs, that's an important factor, but we don't really have a great way of quantifying that. So we weren't really able to include it in the model, which I think is short-sighted. I think there needs to be a way to quantify that particular impact, because I have a suspicion, as well as lots of other researchers out there, that it's going to be an important factor. Expanding our invertebrate data. So initially, when I first started at USF, we really wanted to do a succession study. We wanted to see how an artificial reef develops over time, and are there specific stages within the development that are more crucial to getting it across the finish line, getting it to a state where it can be more useful to natural reef habitat or supplementing biodiversity, that sort of thing. But we don't have a lot of environmental data, or I'm sorry, we don't have a lot of invertebrate data on not only artificial reefs, but even on natural reefs. Most people who go out to do surveys like that want to look at fish. They're easy to count. It's easy to distinguish individuals from one another. The invertebrate data, plant data, all of that stuff is really important to look at in succession, and we just didn't have access to that for that particular study. But I do think that if we were able to get our hands on that, map that succession of those artificial reefs, look at the different stages that they go through, it would be really important information for managers in regards to how they're able to use these artificial reefs as a management tool long term. And then lastly, to expand on the influence of our spatial autocorrelation. So what we decided to do like a week ago, which you guys didn't get to see the results today, unfortunately, but we're going to add it into the paper before we publish, is creating something called a variogram, which basically is going to help describe the spatial continuity of our data. Now, the reason we chose to do this is because a lot of the data we had was repetitive measures at the same site, and that might have skewed our p-values for our spatial autocorrelation, because you're looking at the same site over and over and over. The most repetitiveness that we saw was with Will's data. He was sampling 4 times a year for 6 years. That's quite a lot. The other sites range from twice a year to once a year over 10 years. So still a little bit of repetitiveness in there, which was good, you know, for the rest of our study, but it may have caused a little bit of skewness to our spatial autocorrelation component. So we were planning on running or creating a variogram to help distinguish the level of spatial autocorrelation from that impact of those repeated measures over time. And so that wraps up everything I was going to say. just wanted to thank my lovely committee members, Cam especially, for being so patient with me. I've been here for 4 years, so it's been a long time. All my lab mates for listening to me, having me do my presentation and help read my papers for weeks and weeks on end. My family and friends who are all watching from the Pacific Northwest, and my funding source, NOAA, for funding this MRA Scholar Program. I've been funded by them since I got here. Would not have been able to do this research without them. And the people who gave me their data, I will forever be grateful. Without you guys, Will, Chris, Ted, and Arnaud, I would not have been able to do such a cool study something that I'm so passionate about specifically, and you know, continue to work with, you know, reef data and artificial reefs in particular. So that is all I have. I'll open the floor for questions now. Sorry, deep breaths, everyone. Yes?
Well, thank you. Could you expand on your definition of spatial autocorrelation and how you, in terms of how you use it in your study and research, and how you're going to continue your research?
Right. So basically, we kind of defined for our study spatial autocorrelation. We said, okay, well, if you have 30 artificial reefs or you have 30 reefs in general, and they're all close to each other within, you know, 10 meters, 20 meters even, those are probably going to look more similar as far as their community structure, as opposed to if you were comparing one reef that's 100 meters away from the existing reef. So the closer they are together, the more similar those community structures are going to be. As far as the future, like I mentioned, all our reef sites had repetitive observations. So we weren't able to account for that with the GAM model. So we thought that perhaps because you were repeating the observations, maybe that spatial autocorrelation component was seeming like it was a little more important than it was, just because you're looking at the same site, you know, 10 times in a data set. And so the variogram is really going to allow us to eliminate the impact of the repetitive measure and just look at overall the distribution of species, and if they are, in fact, really reliant on that autocorrelation based on where the reefs are located.
I understand it. So the proximity of reefs and clusters, let's say, within a certain distance, regardless of where they are relative to the Gulf, west or wherever, they're going to show similar characteristics, or am I misreading?
Right, so if you're looking at 20 reef sites and they're all in the northern Gulf of Mexico and they're relatively close to each other, they're going to look more similar, statistically speaking, just because fish move around, organisms move around. So if you're sampling 4 times a year, chances are if you go down once, you're going to see a group of species, and if you keep going to that same site, averaging the observations that you're going to see there, they're probably going to be very similar. And then if you were to just jump all the way down to the West Florida shelf, maybe well, not maybe, the observations are going to be different if you're looking at the species level especially, because the connectivity between those two sites that far away is not very great, because we weren't looking at a lot of highly migratory species or anything either. So does that answer your question? Okay, thank you. Alex?
So you mentioned that you used a few different gear types like the ROV and the drop cameras, and you said you normalized them. What exactly do you mean by that?
So the units are a little bit different when you're looking at an ROV because it's a transect survey versus you plop down a stationary video, takes video for 20 minutes, and then you count the fish. We put them all in a data set together and essentially normalized them, did some calculations so that they don't specifically fit into those units anymore, that they're all similar enough to where the differences in measurement and the differences in unit aren't going to play a huge role in the distinguishing the different reef types. Does that make sense?
So is that just like descoring it, or
It's literally just a normalization function. That's all it was. Do you have any thoughts on that, Cam? I'm probably not describing that the best.
Well, I don't quite remember exactly what you did, but you made, like, among the stationary video observations,
Right. So that's, yeah, okay. 0 to 1 on a scale. That's what we did, and everything fit into that. That was what it is. Sorry, forgot.
There's an age-old question that needs to be answered. You'll run into two kinds of people: those who hate these things,
Mm-hmm.
and those that love them. I did a lot of diving in all those rigs on the northern Gulf, and the question is whether they simply attract existing fish, or do they create more fish?
Mm-hmm. Yeah.
I know what I believe, but anyhow, that's an argument, and
Right.
and none of the statistics will prove one way or the other.
Right. So my literature review that I did at NOVA before I came here was looking at studies that incorporated artificial reefs into the boundaries of a marine protected area, so they weren't able to fish on them. And there are very few studies that have actually done that, but the ones that have actually performed that and published data on it, if given enough time, which time scale we're talking at least a decade for an MPA to be functioning properly, they can lead to more production, but because they're used as a fishing tool in the recreational world and diving, we haven't really been able to get our hands on hard data that suggests that, yes, definitively, they are going to do good things for the marine environment. I think if we had more strict regulations, which is probably not a popular opinion in here or with the school, but more strict regulations on how the fishing community uses those artificial reefs, we'd get a more clear picture of their ability to produce fish. I think in the right circumstances they can, but there are circumstances where they're just attracting all the big guys, and then you go out there with a spear gun and have a nice day on the water. So it's really going to depend on what the management strategy is surrounding that particular reef.
Well, it's an age-old problem that needs to be.
I know. It's like,
You talk to fishermen in the northern Gulf of Mexico, Louisiana, or
Mhm.
Texas, almost all the fishing is around rigs.
right.
All the diving is around rigs.
Mm-hmm.
Now, I don't know what it is today, but in the past, you could tie onto a rig and do all the fishing you wanted, but now because of security reasons, I'm not sure
Yeah.
you're welcome to do that anymore.
Well, I think to your point too, with the rigs especially, and compared to the natural habitat in the northern Gulf, you don't have a lot of vertical height on the natural reefs up there. And on the rigs, you have all of this new spatial habitat underwater. And so I think that it's very possible that they could help to increase the productivity and the biodiversity, but you have to restrict the fishing in order to even get a definitive answer for that for a long time. And that's not a very popular opinion.
Rigs, you can see red snapper all the way
Yep.
to the surface.
Yeah.
Now, if you do your diving on the East Coast
Mhm.
out here,
Not so much.
you'll never be able to dive deep enough. to
Right.
find a red snapper.
Yeah. So I believe they can work, but it's really about them. It's going to come down to the management for that as a tool. Anyone else? No? You guys are easy on me. Thank you.
All right, well, let's thank Elizabeth one more time. And you're free to go. The committee will be back here in 10 minutes, and anyone who wishes